Hi, Emily.
How's it going? You could. I have some company, so I will probably, I might have my video at points, but
That is absolutely fine. I'm just hoping we get at least a few audience members. And if you can concentrate through the distractions, questions are lovely.
She's impressed, kiddo. Lauren's gonna come.
I am. Oh, Lauren, thank you. After all that work today, so grateful.
We will be there for you. I'm looking forward to it. I'm sure there'll be really good talks.
They are, I think, given their slides, going to be really good, and just their performance throughout the course. So I think they'll be good. I'm glad somebody will see them. And hopefully it'll feel like a little bit of pressure for them so that they can get a sense of presenting to people.
Yeah.
It's so hard to make it feel like a rigorous experience when it's all kind of virtual.
Absolutely.
Hi,
there. Okay. I'm just gonna mute the microphone, but we are here and we are excited. So thank you, Jacqueline.
All good. Of course. Thank you. Thank you. Robert can ask questions too if he has any.
I'll tell him.
One girl is interning at a veterinary clinic. That might be fun for him.
That sounds amazing.
Hey, Lauren, thank you so much for coming. Hi, Lauren, thank you for coming.
Like this. I'm excited.
Wait, Mom.
We're doing some Play-Doh here.
Oh, Play-Doh is so much fun. We have done so much over the course of the pandemic.
Does the smell of it bother you?
No.
No, it bothers Chris, but I don't really smell it. Like, it seems like some people it's really offensive. Does it bother you, Lauren?
It's not my favorite.
Yeah, like to me, I barely even notice that. It's funny, but Chris, it gives him headaches.
Huh, that's really interesting. I mean, I know it has a smell, but it does not bother me.
Showing you his Play-Doh cutout, everybody.
Oh, that's really cool, Robert.
Oh,
I like the color too.
Oh, and your Play-Doh scissors. Yeah. Oh,
Those are awesome. You can cut things, but not other things in the house.
They keep eating on my Play-Doh scissors and sometimes snip Play-Doh, but these aren't used to snip here. But my Play-Doh scissors aren't sharp.
That's right. They aren't sharp and they aren't used to snip hair, which is what he said. Did you get a haircut today? Did I give you a haircut? How did you feel about that?
Yeah, but ow, that hurted.
There's just going to be running commentary for a while. I'll just turn the microphone off for a little while, but we are here.
It's okay. We don't have anybody else yet, so. Zoom, it seems like it should make everything easier for us presenting because you don't have to like run around and get the food and everything, but I find sometimes it is actually has some different challenges, like the fact that nobody arrives until moment of, except for my student who's supposed to be safely driving. And I just saw her pop into the waiting room. The one I specifically told it was okay to be late.
Lauren was doing lots of letting in at the symposium of like people who lost their registration and just like constant. There's a lot of layers with Zoom. Yeah, Lauren was quite busy just now. It gets surprisingly complicated.
Hi, everyone. We're going to give it just a couple more minutes. We're waiting on our first presenter here.
I think I see the first presenter sitting next to Emily.
Maybe our most charming guest, but all right. Well, I think we now have our first presenter. And I know Angie would really like to see Tina present and needs to get going. So we'll go ahead and get started. I want to say thank you all for coming. And I want to give just a brief introduction about the course. This is the first semester that we have run it, the symposium just with the course and not with the facilitated program students. But it's a pretty cool course if I don't say so myself. Of course, I'm the instructor, and the course is designed to really help students make meaningful progress towards graduation while engaging in professional development experiences. So in order to enroll in the course, students actually have to submit an explanation of their experience and 3 learning objectives that they then engage with throughout the course. And while some students submit kind of your traditional internship experience, a lot of students will actually go through the course utilizing a full or part-time position that they've had longer term. And while these aren't kind of your normal traditional internship, they are related to their career interests. And then we utilize the course's framework to help students see their experiences through an internship and professional development lens. And then throughout the course and throughout the symposium, we will be using the term internship to describe these experiences, even though they run the gamut of some other options as well. And then I want to just briefly introduce our class. They're going to introduce themselves individually. But this semester we had a really small but very impressive class, and they've submitted some really amazing, well-written assignments. So far, you guys are my top scoring class. And 201, you've submitted excellent CVs, which is usually a very challenging assignment to students. It means that you're able to articulate your experience in different ways. And of course, you've done this all while juggling online learning with your other classes, working in several cases, many, many hours a week at your internship site, and then everything else you have going on. They are also a really diverse class with a variety of interests, both career and otherwise. And this has been really fun for me as the instructor to engage with throughout the course. And I'm really excited that they'll be able to share a little bit with you about all of that today. The symposium fulfills the oral component of the class and is part of their final grade. So how it'll work is we'll have students present in the order on the agenda. And if you guys want to see that, you can see the link in the chat. I put that there. Let me know if anybody doesn't see it. Each presenter will introduce themselves and give about a 5-minute, give or take, presentation. After the presentation, there's time for questions. And please feel free to ask any questions you have either about their experience itself or about the information they presented on. And fellow students, if you have questions, you're certainly welcome to ask as well. And then after we're done with questions, we'll move on to the next presenter, who again will introduce themselves, and we'll repeat the process. So without further ado, I'm going to let Jackson get started. I'm going to stop sharing, and whenever you're ready, go ahead and share your screen and you can begin your presentation.
All right, everybody able to see that? Perfect. All right, so my name is Jackson Worley. I am actually a double major in biology and Spanish. I'm a senior and I'll be finishing my schooling this fall. I did my internship at PRA Health Sciences, which is a pharmaceutical research company located in South Salt Lake, across the street from St. Mark's Hospital. And we focus on performing phase 2 and phase 3 clinical trials for newly developed pharmaceuticals. The company itself is very big. They provide service in more than 90 countries, and there's around, I believe, 60 buildings worldwide. So as I said, we perform clinical trials on newly developed pharmaceuticals, and we try to determine their safety and overall effectiveness before they're allowed to go to the market and also to the FDA for final approval. Their mission, I'm just gonna say it directly from their website, is to develop life-saving and life-improving drugs with our comprehensive clinical development services, including data management, statistical analysis, clinical trial management, medical writing, and regulatory and drug development consulting. And so my specific role in all of this was working in the biology lab. I was processing samples, specifically blood and urine samples. We would receive hundreds a day, and we would centrifuge the blood samples. We would then separate its contents into specific cryos, or really small vials. And then at that point, we would put them into really big freezers that would get down to about -80 degrees Fahrenheit. And then after that, depending on where they needed to go, we would then ship them. And some would be shipped to Europe, South America, wherever they were needed for further bioanalytical analysis. Challenges working here, obviously just time management, self-management, making time for school, and also working a good amount of hours. And the allotment of trivial work. When I first started, I was kind of given the easy tasks, not really what I wanted. I wanted to, get in and start doing everything. But with time, I was eventually given more responsibility, and now I'm to the point where I can walk in and start working. I know exactly what's going on, what needs to be done. So that's nice. And also the repetition of processing samples. Like I said, we would receive hundreds of samples, and many of them would be the exact same, for example, vial of blood, just from a different subject, but it needed to be processed in the exact same way. So with a lot of repetition, it did sometimes get kind of boring. I hate to say it, but it was interesting to kind of sit back and think about actually what's going on in terms of the biology and chemistry that was happening in the lab. And then also receiving little feedback from superiors. Not very often would I be told that I was doing a good job, or that I was being told that I was doing a bad job. None of us actually were told that. I'm kind of the person where I would like to be sat down and told that I'm doing a good job, or I need to improve on specific things. I just like to know what's going on in that sense. What was learned in my internship? I think the biggest thing is the difference from learning out of a textbook and also seeing what was in the textbook that was actually applied into pharmaceutical research. For example, I remember learning about centrifuges. It was interesting, but actually doing it and seeing the contents makes it way better. And the most useful courses and background, I think, that prepared me for this internship was working at Malahay Clinic, kind of gave me some patient exposure. And then also Rocky Mountain Therapy Services, which is a physical therapy clinic, kind of helped me learn the physiological terminology, which was used a lot. And then courses, I would say cell biology and specifically chemistry. Chemistry labs, working with pipettes and freezing, splitting, centrifuges, all of those types of techniques were used daily in the lab. The impact that this internship had on me, it taught me to it taught me that I really actually prefer working with people, speaking with people on a daily basis. I don't like being behind a computer. I found it really enjoyable to have a team of lab members and all of us working on the same thing at once. And we were able to chit chat, kind of made the day go by fast. And ultimately, I don't want to continue working there for too long. It was a great experience. I loved it. But working with, human secretions and specimens, after a while, it does get old. And it can be kind of, makes one apprehensive to go about the laboratory when you don't know what's been on what. But I want to give a special shout out and thank you to Tasha Smith, who is my lab manager, and also to PRA Health Sciences and my coworkers for really providing and maintaining a comfortable and respectable work environment. It's a great company. I can see why they've grown so much. And if anyone has any questions, I'd be glad to answer them.
You mentioned it was challenging not to get a lot of feedback, which you've mentioned before in your papers in passing. But did you feel like the learning objectives you set for the course helped with that at all? Were you able to gauge your work utilizing those, or did they not help?
No, I would definitely say that it helped. I was able to kind of take a step back and analyze my goals and see whether or not I was doing well. And I think I was doing well. I did not make nearly as many mistakes as I did when I first started back in November. So, yeah.
Holly, did you have a question?
Yes, I just wanted to ask you, Jackson, so it looks like you had this lab experience and then a couple more preceding that. Is it something, are you looking to be a researcher? Are you looking heading toward medical school, or what was your interest in having this particular type of experience?
So I actually have plans on applying to medical school this June.
Mm-hmm.
And so I was in search of a job, and I really wanted to try and find something in the healthcare industry. And what interested me most about PRA is you kind of see what's going on in the background. I worked a lot with physical therapists and physicians, and you see them take blood and the blood disappears. You don't really see what happens beyond that, except for just the results come the next day. So what I really liked about PRA is I was able to be in the background and see blood come in, go through its processing, be sent out, ordered, and then receive more like analysis on that specific vial of blood. And I actually really liked that. It kind of taught me a little bit about what's going on.
Great, thank you.
Thanks.
Jordan, did you have a question?
Yeah, thank you, Jackson, for that presentation and for engaging in the class. I guess you kind of answered my first question, which was, how did this internship impact, if at all, your career goals and aspirations? And then also, like, how did you actually find this particular internship opportunity? What was the mechanism you used to connect with that?
Yeah, so like I said, I was in search of a job, and like I said, I wanted to be in the healthcare industry. And I actually know Tasha Smith a little bit. She actually lives in my neighborhood, and yeah, we just kind of bumped into each other, and she mentioned that she works at PRA and that they were in search of more lab technicians, and yeah, it started from there.
Thank you.
Thanks.
All right, it looks like there are no more questions. If I skipped anybody, I apologize. But Jackson, if you'll go ahead and stop sharing your screen.
Yes.
And then Tina, whenever you're ready.
Hold on one second here. I've actually never shared from this computer, so give me one moment. Actually, could you, Jacqueline, could you share that for me?
Absolutely.
Thank you.
All right, just let me know when you want me to switch over.
I will, thank you. So I'm Tina Grimes. I'm a junior at the U studying mathematics with a statistical emphasis, and I work at Health Systems Innovation and Research. You can hop to the next slide. So HSIR is a program that partners locally and nationally in prospective and retrospective studies. And that just means in general that we will either look in the past, we will find patients and look at their past medical history, or we will test things currently. So Jackson, he works somewhere where they test the safety of drugs. One of the things that we will do is occasionally we'll partner and we will test whether drugs work on something that maybe they weren't originally made for. For example, we're working on a study right now that's testing whether hydroxychloroquine has any positive effects in treating COVID. There's also studies, we've done studies on flu shots. We work really hard at HSIR to improve healthcare. So my key responsibilities currently as a research assistant are data entry, which is pretty self-explanatory. I just put that data in that we get. There's also query resolution, which is under data cleaning. So that's essentially, they'll come to me with a problem like this data is missing or we have a data disparity, and I find where the origin of the problem was, and then I fix it. I also do chart reviews. So chart reviews are when you'll look at a patient's health record. And for example, we might be looking for specific health history, or we might be looking for whether they've been hospitalized. I also develop study procedures and data capture systems. So study procedures would be, for example, if you're hired on here, there's something called like a workflow or a MOP that tells you what to do, where to go, who to talk to, and I write those, most of them. And then data capture, for example, would be fields, like instruments, like I'll look and I'll see, okay, we need to decide when recruiting patients, whether they're recruited into this study or this study. And so I'll go and I'll make a form that will branch out depending on when you're talking to that patient, what you put in. Next slide. So again, with the workflows. So once we moved remote, it was really important that our workflows were very detailed. So I got to help with pretty much overhauling all of those and adding new ones. And they were, one, very beautifully formatted, if I do say so myself, but also really thorough. So that was nice. And I'm also co-managing data on one of the largest outpatient COVID trials in the US, which sounds very fancy, and I'm pretty proud of it. So that falls under the data cleaning and that query management. I'm helping make sure that that query is actually, that data is actually clean and good to go for when it actually heads to analysis. Some of the challenges I've encountered, this first one was a little more when I first started out, which was a while ago, but it was an entirely new knowledge base to draw from. So for example, I didn't know what an IRB was. I didn't even know what a workflow was, but I just took a lot of notes and I still write down pretty much everything. And kind of, you have to learn to trust yourself when you encounter a lot of new things. And we're a very small team, so I do have a very large workload, especially considering that I'm part-time at this job and then full-time at the U. But it was just a good opportunity for me to learn how to ask for help and stop saying yes to everything that I was offered. Next slide. So I learned, well, I'm still learning a lot at my job, but some of the things that surprised me the most, when I first applied, I really thought that, oh, I'm going to be learning all this fancy healthcare stuff and maybe stuff about labs. But it was really the small skills that I feel like I've been taught really well. For example, like email etiquette and just kind of learning how the office office environment works, how you work in a team. Of course, that's not to say that I didn't learn a lot of good information about healthcare research. I really got to see what it was like to get like down on the ground and learn that information, like what it's like to talk to patients, what it's like to try and get these studies off the ground, all the different pieces that have to work together, all the effort you have to put in. And I think that'll be really useful for in the future because I'm in statistics and may go into, you know, data analysis. It's good to know what it's like to be on the other end of that data that you're looking at and how it's collected and what might affect it. One of the things that was really, oh, could you hop back? Sorry. Yep, right there. One of the things that was really useful to me, again, was that I'm certified in like Microsoft, which sounds like kind of a silly thing to be, but it's essentially just proof that I'm good at Access, I'm good at Excel, I'm good at Word. And you know, you use Microsoft every day, and I use Excel a lot to kind of clean up the data because I'm not working. I work with maybe a maximum of like 15,000 records. And so Excel can be really useful if you're looking at a small bit. Maybe I just need to look at who's withdrawn. Maybe I need to clean out patients with heart disease. So it's really useful to just have those tiny skills. Next slide. So like I say here, so working at HSIR reaffirmed my interest in the healthcare field. I wasn't sure if that's where I wanted to end up. I was half and half between maybe going into IT, where I worked for a little bit, or going more healthcare because I really wanted to work somewhere that maybe had a future forward positive like impact. And this job also really taught me what to look for just in those jobs. For example, I've learned that it's really important who you work with, not necessarily always what you're doing. But again, I've talked to some people, and thanks to this job, I'm considering maybe becoming a biostatistician, which is pretty cool because I struck it off my list a long time ago, but now that interest has been rekindled. Next slide. So I just want to say thanks to Ainsley, who's my supervisor, and my wonderful coworkers and HSIR administration, who has had to deal with paperwork of me flip-flopping from full-time to part-time to work study to not work study. I'm really glad that I work here, and I think I've learned a lot, and it's just all the people are great. Thank you. Anybody have any questions? Go ahead, Holly.
Hi, thanks for that. That was really interesting. So I'm curious about doing statistics out there in the real world. And you mentioned at the end there that you were thinking about being a biostatistician. And I think you said you were a junior, is that correct, now in college?
Yep.
Yeah, so if you were to choose that path, would it be something where you would now need to take some courses in, biology, for example, to get a little bit of a background so that you can understand that data better? Or do you feel like your statistics background alone would help with that? I'm just not really clear on how, how do you apply statistics in the real world? Do you need to have like a different content focus in addition to that?
Sure, yeah. So I actually have kind of kept my foot in both doors, kind of looking at what happens. So if you go into biostatistics, you usually have to get a master's. So I actually talked to Lauren, who is in the PHS department, which is what our department is under, because she's the program manager for biostatistics, I believe. Biostatistics. So I talked to her and asked her, like, should I take some biology classes? Should I not? And she said that they try to be really holistic and that it could be helpful, but that in general, just as long as you have a solid statistics background, that can help you and you can learn the biology essentially later. Of course, it's different for every program, but.
Mm-hmm. That's great. Well, it was really interesting. Thank you.
Thank you. Jordan, go ahead.
Thank you, Tina. That was really interesting. I guess I was intrigued by you because you mentioned it a couple times that part of your job is cleaning data. And I'm really curious about what this looks like. So sorry, this is a little bit of like a, sort of a specific question, but I'm kind of curious about what this looks like in the context of sort of a regulatory environment, right? Where you're working under IRBs and there are all these, procedures and guidelines around data, and particularly with things like clinical trials and whatever. So what does sort of data cleaning mean in that context? And like, how does it play out in, when you're doing it in that way, with regulations and things like that in play?
Well, the regulations are part of why we make sure the data is so clean, because we don't want there to be mistakes. We don't want to, for example, violate consent. That's really obviously, for a lot of reasons, something you want to do. But in general, with data cleaning, a big part of clinical studies is essentially just write everything down. Write down when you call the patient, write down what you talked about, within reason. So data cleaning often just looks like finding the mistakes, and either correcting them. For example, if it's something small, like just a mistake in data entry. For example, I catch a lot of they messed up the dates, because depending on the database you're in, it's a tiny thing, but sometimes the day comes first, then the month. So sometimes you'll get, this patient was consented in the future, and we know that's wrong. So we just check and we can confirm. But sometimes you do find actual mistakes, and that's why there are regulations, because then you can submit that. You can let, IRB or coordinating center know that there was a mistake, to both make sure that you can set up new systems that make sure that mistake doesn't happen again, and just make sure that you can always go forward and make sure that your data is both honest and easy to interpret.
Thank you.
I'm actually curious, Tina, what made you take biostatistics off the list initially?
Grad school, actually. I just wasn't sure if I wanted to do it, because I mean, obviously you guys are either in school right now or you've finished it, and it can just be absolutely exhausting. And I was just like, I don't know if it's worth it, especially because I did have a couple people that warned me that, like, you don't want to stay on that path, like you don't want to lock yourself into one specific like field. And some people warned, like, oh, like there's less opportunities for that reason. But now maybe I can go to grad school. It looks like maybe Emily, you have a question?
Yeah, I did have a question. I was curious. It sounds like you've been in this position for a while, and I was wondering what the learning curve was like. What was it like when you first started out and you were trying to figure out all these pieces and your role and how to juggle your workload? Did you feel fairly comfortable after a month? Did it take longer? What was that process like for you?
It was really a trial by fire. Again, we're a very small team. So the second I was hired, it was essentially like I had to be on the ground and like actually contributing. I couldn't give it a specific timeline. I think it took a long time, especially because HSIR, you're not doing the same thing. I haven't been on the same study for like all the years I've been here. So you'll find your stride and then the study will end. But like Ainsley, my supervisor, and my coworkers, like you just have to work together. And kind of, if you're confused, you have to ask, because someone else will know. So it can still be a learning curve at times, but I think but I think you get used to it, and it can be kind of fun to constantly have things changing.
Thank you.
Does anyone else have any questions?
All right, well, let's go ahead and move on to Jeremy.
Okay, I'm just going to the link right now. Okay. So I'm Jeremy Sarton. I'm a senior. I'm graduating next week, and I'm a physics major, getting my emphasis in applied physics. For the past 8 months, I've been working as a mechanical engineering intern at SnapBlock Industries. So what is SnapBlock? SnapBlock's a distributive modular flooring. It's basically indoor and outdoor sports flooring, or as if you look in the picture in the top right, it's we also do garage flooring. That's our race deck tile. Basically, it's polypropylene and propylene plastics, so very durable, lasts a long time, and 100% recyclable. So my job at SnapBlock was making sure that these tiles were produced as efficiently as possible, while also making sure that they performed as efficiently as possible. If you look at the picture in the bottom right, that is our ASTM force reduction test. And we just use that to measure if the tile can take a lot of force over time, or if the tile can take a lot of force at once. So it actually means a lot for making sure these tiles are good for your ankles, knees, hips, back. It's a lot of running and jumping around. So it's important that they can take a lot of force. So some of the challenges that I had were last semester, I was only working there 1 day a week. And when this semester started, I had a lot more free time. So we increased it to 3 days a week. And that meant a lot more job responsibility. I was given a lot more long-term projects instead of short-term projects that can only be done in a day. And it was just really difficult getting used to switching over my mindset for being just 1 day at a time to thinking over weeks or even months of project length. Another challenge is learning all the ASTM tests. A lot of times we would just get instructions saying, here's a piece of rubber, come back to me with the hardness of it. So it took a lot of times to figure out what device actually measures hardness, which is a durometer. And another challenge that I had was figuring out the tests that were designed by past interns. So all ASTM tests have procedures, they have lab manuals, usually 40 or 50 pages that just tell you exactly how you're supposed to do the procedure. But when you have tests that are designed by interns, a lot of times the procedures get overlooked. And I have a video. I was having some difficulties playing it earlier, but can everyone see this? So this is our rolling load tester, and it's a linear actuator hooked up to an Arduino, and it takes that 300-pound force over a wheel, and it rolls over a tile back and forth 200 times over 5 sets. And after each set, we measure the deformation in the tile so we can see how well it holds up with a lot of weight over time. So some of the things I've learned, having a background in this industry, I've actually worked in the modular flooring industry back in Georgia, my home, since I was 15. So I was able to just jump right into this job knowing all the details of the industry, and I was able to just be effective right away. Another thing that helped me be really effective too was the lab classes that I took at the U. They really prepared me for procedural work, specifically the physics and chemistry labs that I took. Really taught me what it was like to go through a procedure and come back with standardized outcomes. And then one thing that I think I'll take with me from this internship is really my proficiency in Arduino. I've used it in a few classes at the U, but since having this internship, I've used it probably weekly, and I've just gotten a lot better at that language. So lastly, this experience, it was amazing. It taught me so much about the engineering industry as a whole. I'm really appreciative for everyone at SnapBlock for giving me this opportunity, especially Jeremiah Shapiro, my supervisor. as well as Carrie and Jorgen Moeller, the owners. They really looked out for me and helped me out. I would also like to give a shout out to my fellow intern, Greg. He's really made working there a great experience. Yeah, so any questions?
Greg's an engineer, correct? Engineering student, Jeremy?
Excuse me? Oh, yes. Greg is a mechanical engineering student. Yes.
How do you feel like your physics background complemented that?
I think it worked really well because he was very structured in what he did. He also has a framing background, so he liked to do things very specifically. And I feel like with my physics background, a lot of what we did was problem solving. So I was able to bring a unique problem solving background to his very structured mechanical engineering background. I think it worked very well. Mackenzie, you have a question?
Yeah, I was just wondering what your favorite part of your job is, or your internship.
My favorite part, I would have to say working with my hands. I really didn't like to work behind a desk, and this internship gave me the opportunity to design a project in a day, order all the parts, get there 2 days later, and then build for 8 hours. And it's just something I really like to do. Holly, you have a question?
Yeah, so I'm fascinated that you were given the opportunity. It sounds like you were given kind of a tough job, like trying to figure out what machine to use and what was the most effective. I mean, that seems like a lot of responsibility for an internship. So did you get a lot of feedback and support doing that? Or was it kind of, I think you mentioned trial by fire in some cases, but that sounds intimidating. I'm just curious, can you talk a little bit more about that experience?
Yeah, of course. In a lot of ways, it was trial by fire. I could come back with data that maybe they didn't like, and it would, instead of saying, what did you do? It was just go do it again, write everything down, and then come back to me if they're the same results. But I would say a lot of time, me and the other intern, we really looked out for each other. So if I didn't know what to do, I had no idea what a durometer was day one. And that's a lot where the other intern comes in, because where I lacked in my knowledge, he usually made up for it, and vice versa. So we were able to just work with each other a lot and make sure that we were putting out quality work.
That's great. Sounds like a confidence building experience.
It was very much so. Jordan, you have a question?
Yeah, thank you, Jeremy, for that. So I guess I have a couple questions if we have time. I guess my first question is, you kind of mentioned that one of the things that this revealed for you was that you really like to work with your hands. And I'm kind of curious about how, in what other ways has the internship affected you in terms of your ideas about next, what you're going to do next, or what you want to do long term?
That's really interesting. So I would actually say that this class had more of an impact on that. So this class, I was given the opportunity to interview someone, and I chose a professor at Colorado, and he basically, we sat down and we talked, and we went over what I do like and don't like working with my hands and not sitting behind a desk. And he basically walked me through how academia and how doing research out in the field can actually be a lot more, or just like a lot more what I'm looking for. And, also through this internship, I kind of just learned that maybe engineering isn't exactly where I want to be in 5-10 years, but maybe somewhere where I can conduct my own research and I can choose my own projects. I think that's something more in the line of what I want to do going down the road.
That's interesting, and actually that was going to be something I was going to sort of try to convince you that research also feels like that. But I wanted to. The other thing that intrigued me was that was your discussion about how intro labs, I think, like in physics and chemistry, that those were important for you in that they helped you learn how to sort of follow a procedure to kind of get reproducible results. As far as that's what I think I heard you say, something like that. Mm-hmm. which is really interesting in this context, right? There's a lot of work that's being done at the U, but also around the country in developing more inquiry-driven labs where they're more open-ended. So they actually don't have specific procedures. I'm involved in some of this work here at the U, for example. And so I'm just really curious about you as a student, sort of say, like those sort of procedural, like learning procedures may kind of go away, or at least be sort of minimized. And because like, I guess we've been thinking that as faculty that like, maybe it's more valuable for students to learn how to think on open-ended problems and like troubleshoot and kind of go through more of a scientific process than a procedural one. So just kind of curious about your thoughts on that.
I think that'd be awesome. That'd be really helpful. A lot of times at our jobs, we didn't have any procedural methods. If he told us to get our supervisor, Jeremiah, told us to get the hardness of rubber, but we didn't have a durometer, we were spending the day basically inventing our own durometer that we could just use comparable results. So I think that would be a great way to get students to basically figure it out for themselves. I think that's great.
Good, thank you.
Mm-hmm. Okay, is there anyone else?
I think that's it. Thank you, Jeremy. Alexander, you're up whenever you're ready.
Yeah, can I just ask if you can share the screen for me? My computer just shut down and this other one doesn't want to open me PowerPoint.
No worries. Give me just a moment here to bring it up.
Yeah, sure, thanks. Okay, thank you. So, hi everyone. My name is Alexander Lukic. I'm a senior and majoring in biology. As Jeremy, I will graduate next week. Yay! And I did my internship in a company called Biofire Defense. Next slide, please. So what do they do? This company is a company that works for the Department of the Defense and the government, makes biomedical products for disease identification. As they say in their website, their mission is to speed up medical results, help people stay healthy and make community more secure. And we are working with a bunch of different organisms and diseases like COVID-19, since it's the newest one, but we have other organisms like Yersinia pestis, that is the common plague, or like West Nile virus, a lot of different deadly viruses. So, as I said, the products that they're making are for the disease identification. And since they're working for the DoD and the government, only like the military can use these products. They have like a sister company called Biofire Diagnostics, and they are specialized for different other diseases, but the rest of the people can use their products. Next slide. So over there, I was a research associate. I was an intern, and I work with the analytical group. So the duties for this group were to test different microorganisms in different ways and for different reasons. Like, for example, I cannot share too much since they're pretty strict about their privacy. But I can say that like I work with different experiments with the COVID, testing them like with if their products are able to be tested with sputum, blood, saliva, all different type of medias. And we were, as you can see, these images, we were working with these pouches that are made from Biofire Defense, where when we inject our samples, there were different types of pellets inside that help to react with this sample. And there was a different processes that help to break up the molecules, start to reproduce a lot of RNA or DNA, depends from what we are looking. And all these samples were going inside to this little black thing that you can see, where inside there were different RNA or DNA, And with the software that they had can read which organism was detected in that sample. So as I said, I was working with this analytical group. We were doing all these tests and data verification. So basically, when we are done with our experiments, we were just analyzing the data that we got, see if there is any error. If not, start to write a report and send that to the FDA or DOD. Next slide. So the challenges I faced, there were like a few. At the start, I had difficulties to understanding what they were talking about, since it was like a lot of technical words that I never heard, since I was coming from like a manufacturing job. This was my first job inside a lab. So I, like, for the first 2-3 weeks during the meeting, I was just staring at the desk. I don't know what they were talking about. And another challenges was to remember all the security rules that we had for avoid contamination, since this lab is by BSL2, so biosafety lab level 2, which it means we are working with diseases that they should be dead, but not always, because there were a few times that they found out that the organisms are still alive. So they always want to be sure that we are not contaminating other stuff. So they had a lot of rules for avoid this contamination. And another challenge that I had, it was to write and read these lab reports, since English is not my first language, and it was pretty hard for me to read these like hundred of pages and write them down and try to understand what it was written on this paper. But after 2-3 weeks working there, I start to get used and start to understand better what they're saying, what they were talking about, what they want to do. So at the end, it started to go like downhill for me. And right now I don't have any of these problems. Next slide. So what I learned how to work in a professional lab. The only time I work in a lab, it was at the U or my university in Italy. I just work over there, and it's pretty different from school and lab, like real job, because at the U, it's everything like easier. Plus, if you always will get the result that you want because it was tested so many times, while in the real world, you will never know what will happen. If for example, in my case, if the sample, it was already positive, and then can change all the results from our experiment, or all of these like problems that can happen. I start to understand better the world of microorganism and the effect of the diseases in the populations. So I have to learn like what these organism make if in contact with an animal or in contact with a person, since they have a different effects. And I understood how like the researchers are able to identify these diseases and these microorganisms. Before watching like the movies talking about like diseases, they were just saying, oh yeah, we know that it's COVID. And I never know how they were able to identify those. But right now I understand how like all the process behind and how it works. Like the classes that helped me a lot for this internship were biology, microbiology, lab, and math. My math is pretty bad still right now, but the few things that I know that helped me to work, because we have to make in calculations for our volumes that we, how much sample, like, or in the sample we have to spike with different diseases, how much of that organism we have to put in the sample. So math, it's helping us for understand better like the right volume to put inside. Next slide. So this internship helped me a lot to understand what I want to do. Before this internship, I wasn't sure if working in a lab was like the right choice for me, because I was afraid to be too much clumsy to work with these instruments that they're very delicate, and that this job can be very repetitive and annoying. But hopefully it was wrong. I'm not so clumsy as I thought. And there is like, it's not repetitive. Like, as I said, every time you are doing an experiment, you will never know what you're gonna have. So it's always like a new surprise every day. For example, what we want to work today, what is going to be the results. If these results is going to be like this one, what do we have to do after what we got? Or if the result will be another one, we have to start another plan to do like continuing these experiments. So it's not annoying. The only things like I can say it's a little annoying when we have to data verification, because it's just a bunch of numbers and data that we have stare for a few hours in the computer. But hopefully that is like the minor part of the job. Probably I will continue to work in this field because I like it very lot. Next slide. So I would like to thank Biofire Defense for giving me this opportunity to working with there, my supervisor, Jared Helm, and all my coworkers, because they let me to do this new experience, and they were very patient with me since I asked a lot of questions, especially in the start, since I didn't know what I was doing. They were very patient to me and answering me all these questions, give me feedbacks, and everything that I needed. They were always helpful for me. Next slide. So that's it. Do you have any questions for me? Like Holly, I see you like raise your hand.
Excuse me. I have to ask, did you actually work with the plague?
Yes, a few times.
You did? Okay. Yeah. So yeah, it sounds like you were in a challenging environment. I'm curious though, you do a lot of testing and analyzing. Did you come across times where the results you were getting weren't something that you expected? And if yes, like what do you do in that case? Do you have to rerun the test to verify that you got these weird results and then figure out why, or what was the process?
So depends from what was the problem that we got these unexpected results. Few times like these pouches like leaked, or the machine that was running the PCR process have like some problems, and that gave us like a failure. If that was the case, if we just run again the same sample again with a different pouch and a different machine and see what we got. Usually they always work, but it happened a few times that the samples that they were supposed to be negative, they were positive for different diseases. Since these samples that we got, they from clinical sites around the world, like Sierra Leone, Georgia, or other parts of Africa. A few times we got that, especially the African ones, they have some diseases that we weren't expecting that. And in that case, we had to throw away the sample because it wasn't helpful for us, and start again a new experiment with the same concentration that we were using with another sample. Or another thing that can happen was if we spiked the organism inside the sample and it wasn't detected, that probably because the limit of detection was too low. So we had to change like the amount of organism that we spiked inside the sample and restart again this experiment and see what we got from the results.
Wow. Well, that sounds really interesting. I can see why it didn't become repetitive. Something's always different. Yeah, that's great.
Something always happened.
Yeah, thank you.
You're welcome. Jordan, yes?
Thank you, Alexandra. That was great. Really interesting experience it sounds like you've had. I guess I was a little bit curious about how the process that it seems like you must have gotten some sort of security clearance. Yes. And I'm curious about like that process and how long that took and what that felt like. And I know you mentioned that like you can't, there's lots of intellectual property issues, so you can't like, and also probably security issues, so you can't talk about too much in detail on like in this sort of talk, but also just, I mean, what was the process like to get security clearance for you?
To be honest, since I'm an intern, I didn't get through any process because usually I'm not touching stuff like that can be shared, that cannot be shared, so they are secret. So usually they don't give me to work with those things. But as I understood from my coworkers, they were gonna ask you like a few questions, And when they see that you're pretty trustful, they will give you this clearance. But usually all like the scientists, so our bosses, usually they have these kind of problems. We are just working, and the only thing that we cannot share is like the results from our experiment, and try to not talking too much about with other people, that because since it's working for the government, there's always like that threat that probably somebody can sell to a different country and give problems to the US. So yeah, hopefully, as I said, we are not working with that kind of stuff that is have to be secret.
Right. Thank you. And then another question. You mentioned at the beginning, the first 2 to 3 weeks, that you just kind of felt like you didn't even really understand what was happening, like the discussions, like almost a different language. How did you sort of stay positive and motivated during that time and not get discouraged? I mean, I know that like that could be like, I mean, you might say that that's not a hugely long time, but you didn't know that it wasn't going to be a long time until you sort of caught up, right? When you started. So
to be honest, I was discouraged, especially the first week. I was thinking I will never be able to understand what they're talking about. But hopefully, since like my first 2 weeks, I didn't do too much. Since I have to learn, I spent most of my time on Wikipedia and reading their old reports, so I can understand like what they were wanted to see, like what was the process they wanted to do, or like from the older experiments, see what were the results. So trying to understand what we're talking and what the result that they wanted. So like the first week I was spent with that, and the second week, as I said, my coworker were very patient with me. So every time I asked questions, every time I didn't understand, and they were very patient for me explain everything. So as I said, in 2 weeks, 3 weeks, I started to understand what they were talking about and what I was supposed to do.
thank you.
Thank you.
All right, thank you so much. And I think, Mackenzie, we are ready for your presentation.
All right, can you hear me? All right. So I'm Mackenzie Woodrum. It's actually my first year at the University of Utah. Sorry, my dad. One second. Sorry about that. The struggle of living with your parents still, but anyways, it's my first year at the University of Utah, and I'm a bio major. I want to be a veterinarian, so I did my internship at Lehigh Animal Hospital. At Lehigh Animal Hospital, they're just a normal animal clinic, so we do cats and dogs. Our goal is to help as many pets as efficiently as possible. Sometimes we get super backed up, but our main goal is to just help as many pets as we can. Some of my responsibilities, so right now I'm a part-time worker, so I get there halfway throughout the day, and they're normally finishing up surgeries, so I help recover patients. Sometimes I'll switch out and take vitals, but mostly I'm just recovering them, making sure their vitals are good, taking their temperatures, making sure that they're stable. My main responsibility is dealing with the rooms. So rooms can vary from basic vaccine appointments to the dog having problems and the doctor not knowing what it is. I also deal with lots of medication with the rooms. So after the doctor goes in and assesses the animal, then he'll tell me what to prescribe and to pull up. Sorry, one second. All right. Sorry about that. And then, I also do nail trims there too. And my biggest accomplishment is probably drawing blood from a forelimb of a dog. We draw blood from the forelimb of a dog to run heartworm tests. Heartworm larvae are carried by mosquitoes and can inject the dog by like stinging them. And then if the heartworms do get to the heart, there's this complicated process in which we give a couple injections and have to watch them. But with most of these heartworm tests in Utah, they do come back negative. It's more common in the South and the East, but they did do a study and they're finding heartworms to be more common in Utah now. Some challenges that I faced when I first started working there, it was pretty hard to kind of get the hang of listening to dogs and cats and getting their pulse and respiration right, because you can't really tell them to stop panting or stop sniffing everything. And when they are panting and sniffing, you can only hear their lungs and not their heart. And then right now I am working on drawing blood from the jugular. It is a very difficult process in which you have to hold off the vein kind of like at the bottom of their neck. Don't worry, it's not an artery. Their arteries are in the back of their neck behind muscles. In comparison to human, the dog jugulars are about the size of our veins in our arms. We do draw blood from the jugular when we're running blood tests, when either the dog is too old and we need to make sure, just get a general reading if they can handle surgeries, and also to help with diagnosing the patient. And then another thing that I am working on is placing a catheter. It is very similar to drawing blood from the forelimb, except you have to keep the catheter in the vein for the whole time. When pulling blood from the forelimb, you hit the vein and then you pull. With placing the catheter, you have to hit the vein and then stay in the vein as you're proceeding to place the catheter and then push it all the way in. It is a tricky process, and I've only done it once, but I've only tried it twice. So with time, I think that I can place lots of catheters and draw blood from the jugular. The benefits of being able to do this internship at Lehigh Animal Hospital instead of doing a class on campus is working with animals, because at the University of Utah, they don't do a lot with animals, and I do want to be a veterinarian. So it's really good experience. I got to handle crazy animals, but mostly good. And another benefit was drawing blood from animals. So you can't draw blood from an animal unless you're under the direction of a veterinarian or under the supervision of a veterinarian, or so I've been told. Experiences that have helped me get this position was I did do a volunteer work at Saratoga Springs Animal Hospital. And then, although many people don't think of it, but there's lots of cleaning that is involved with this job, and my mopping skills are pretty good. This internship has impacted my career goals by getting me hours. I need veterinarian hours in order to get into a vet school. So this is a really good experience, because vet schools, they don't just want anyone. They kind of want people to have experiences in this situation before they do get accepted, because there are 7 applicants to every 1 spot for vet school. I also like to thank all my coworkers and the doctor. That's Aaron Chamberlain. He's the only doctor we have right now full-time. We do have other doctors that do come in part-time, but he's hoping to get another doctor full-time. And a special thanks to all the dogs and cats that I have practiced on. Are there any questions? Yes, Emily.
Hi, yeah, thank you. That was really neat. I was curious if you've noticed, it sounds like you've been working at this clinic for a stretch of time, and I'm wondering if they have seen a big increase in traffic and demand during COVID. You know, everyone's getting pets, everyone's more concerned about their pets. I'm curious if you've noticed any patterns in that regard.
Yeah, so it's been about 1.5 years since they first had the boom. I've only been working there for about 6 months. But they did notice that everyone was staying home and seeing lots of problems with their dogs. We had one guy come in with his male dog, about a lump on his stomach, and it was just the dog's nipple. So they are noticing lots of things. Yes, Holly.
So are you a vet tech now, or is that okay? So do you have to get a certification for that?
No, so I actually did on-site training for mine. So basically all my coworkers trained me and helped me out. In Utah, you don't need a certificate to be a technician. It is harder to get a job because lots of places won't want to train you, but I was very lucky to get this position.
And so it sounds like you worked in another animal hospital too, but is it what you expected it to be like? So it sounds like you're still solidly like, I want to be veterinarian. So does that help kind of clarify what day-to-day life would be like?
Yeah, I didn't realize how many crazy cats and how many crazy dogs try to bite you every day, but it's pretty great.
Or how many crazy owners don't know what a dog nipple looks like.
Yep. There's lots of those.
Well, this was great, Mackenzie. I think I'm curious about so you said you're a first-year student, right? So do you anticipate that you'll continue this in a position or at this, or maybe, or are there opportunities for you to take additional positions if you stay in this clinic for a while? Like, do you have plans for that?
Yeah, so there's not really many positions at our clinic. It's really small. I could be a head tech if I really wanted to, but that's not really the plan I'm gonna go with. So what I'm thinking right now, I want to work here for about 1 year because a year experience is basically what other clinics want in order to hire you. But I'll probably work here until next summer. And then next summer, I want to do a study abroad in Africa with animals, like larger animals. And then when I come back, I might continue working here, or I might volunteer at the zoo or work at Wasatch Exotic Animals just to get more of a variety before I go to vet school.
Like an awesome plan.
Thanks. Are there any other questions?
Okay. Well, thank you all so much. I think the presentations were wonderful. And thank you to everybody who attended and asked great questions of our student interns. Congratulations to those who are graduating. I know that's super exciting. And then I know we're past time, so I'm going to let you guys all go.
Thank you.
Have a great rest of your evening.
Thank you,
Nice job,
Jacqueline,
everyone.
for running an excellent course, and thank you, students, for all the amazing work.
Have a good night, everybody.
Bye-bye.
Thank you.